So, hello everyone. I'm Maushandra, and today I'll be conducting the first problem solving session for the course Tissue Engineering. So, I'll briefly introduce myself and also I'll briefly tell you guys about my background. If you guys want, if you guys feel free, so I would also like to understand your backgrounds as well, since we'll be meeting for next 7 weeks. So it would be nice to know a bit more about you. Okay, so let me start with myself. I am a PhD scholar in the Department of Chemical Engineering, IIT Kanpur, and I am working in the field of tissue engineering for my thesis. So I work mostly with polymers, and I am experienced, like I have gained experience in working with polymers and using different biofabrication techniques where polymer is the raw material, like 3D bioprinting, electrospinning, freeze drying. So these are the techniques that we use to process polymers to get a desired shape onto which we can seed cells and ultimately use it for tissue engineering applications. So if you like to know more about my research, you guys can have a look at my website. The link is given on this slide. Also, if you have any queries, feel free to ask me during the sessions, and also you can post them in the discussion forum as well. So, every week on Saturdays, 4 to 6 p.m., we will meet and we will discuss your queries. And I have also prepared some questions which are related to the week's content. So, it will help you understand the content much better. The link is with you, right? And all the sessions are recorded. It will be made available to you by the PMRF team. It is going to be uploaded in my YouTube channel. You guys can check it out at Mochandra PMRF IIT Kanpur. Also, the notes that I'm going to use during this session, they will be also made available to you. And towards the end of this course, I plan to take one revision session as well, but that will be after week 8. Okay. So the meeting link that I have given you, the platform, it doesn't allow you to write anything in the chat. So it must be the chat option must be muted for you. So if you have any queries, you have to just unmute yourself and ask. Or if you want, you can also raise your hand. I can call on you and you can ask your queries. But the thing is, you have to interact more and more. So quickly, if anyone feels motivated, you can introduce yourself to the rest of the people here. Many have joined, almost 12 people have joined. That's nice to see. Anyone? It's up to you voluntarily. Like anyone can unmute yourself and introduce yourself.
Hello, am
Hello.
I audible?
Yes, yes, you're audible.
Yes, so I'm Gauri Shripriya. I'm doing my post-graduation in PGIMER in medical biotechnology. Yeah, so that's about me. I'm in Chandigarh right now.
Great, great. Thank you. Anyone else?
Hello,
Hello.
I am Rupkota Shade Khan. I am doing my MSc Masters in Biotechnology from Purdue University.
Oh, great. Anyone here who is not from bio like or life science background, like any other background like material science, nanotechnology, or any diverse field, anyone? Oh, great. It seems that all of you are from bio-based background. So maybe that's why you guys have chose to enroll on this course to know more about this field. But the interesting thing about this field that it's an interdisciplinary field means even a chemical engineer or an electrical engineer can find a research problem in this area and work on it. So it's not just limited to those people who are only studying life sciences or biotech. So that's a great thing. So let me just quickly recap the course content that you guys have gone through for week one. So first you have obviously studied the definition of tissue engineering, right? So in very briefly, what is tissue engineering? It is basically an interdisciplinary field where the principles of life science and engineering both are used together in order to make something useful, to have a biological function. That useful thing should have a biological function that you can use for purposes like improve tissue function, repair tissue, or regenerate it, right? So from this definition only, we come to know the 3 R's of tissue engineering. I request you all to please mute yourself. Okay, if you have any queries, then only you unmute and ask. All right, Ramaya, so please mute yourself. So as I was saying, from this particular definition only, we come to know that there are 3 R's of tissue engineering that gives the goal to this field, where the researchers who are working, are working towards these 3 goals: to regenerate damaged tissue, to repair it, or to replace it altogether. Okay, so how do you make an artificial tissue? So tissue engineering, if you translate it, we are trying to make or engineer a tissue artificially in a lab. How do you do it? So you need raw ingredients first. So what are those? You need 3 things. First is the cells, obviously. The cell, you have to find out from which source you want, what type of cell you want, right? And then cells themselves, they cannot grow without a substrate, a solid substrate, right? So that is why we need a biomaterial. Okay, and when cells and biomaterial together sometimes is not enough, in order to direct the maturation of an tissue towards a particular organ, suppose, we also need to supply some signaling molecules. This can be some type of growth factors. So these 3 ingredients are required for making an artificial tissue in lab. And we'll come to later on what type of cells you can use, what type of biomaterials, what are biomaterials, like we will like briefly touch upon during this session through questions, through solving questions. I would ask you this: what is biomaterial? What do you understand by biomaterials? What type of biomaterials should you use? Like, I will ask you these questions. So these things will get clarified when we discuss later on. Then we also saw what are the classes of biomaterials generally used for tissue engineering, right? So there are polymers, metals, ceramics, right? Then sir also told about carbons, right? Carbon nanotubes, graphenes. There are also mexines that have started coming up. So these are all carbon-based materials. And when you make a composite out of it, like you combine polymer and ceramic, polymer or metal, so these are composites. So these like this, there are 5 classes of biomaterials, and each particular type of biomaterial class has its own purpose. So when you're trying to make an artificial tissue, you first have to think what is the purpose, where, to which organ you're targeting. What Then you think what is the mechanical property of that particular organ. Then what other So that we are trying to mimic, right? Artificial tissue is trying to mimic the properties of that particular organ so that we can regenerate, repair, or replace. So we have to think in that direction, and accordingly you will be able to choose the biomaterial of interest. Then there are various cell sources that are present. So you can directly derive from the patient, that is autologous cells. You can take from a relative of that patient, allogenic, or you can take from an animal, right? Mammalian animal, xenogenic, or you can also take it from a twin. So if you know the patient has a twin, so you can take cells from that person as well. It is called syngenic. So in this way, there are various cell sources that are available. Then Yes, you've identified the source. Then what type of cell is needed for that application? Do you need a differentiated cells? Do you need stem cells? Do you need like any embryonic stem cells are there? Induced pluripotent stem cells are there. So depending on your application, you would have to select the cell type. And as well as you can choose a cocktail of signaling molecules in order to control the maturation process of your artificial tissue. So this is like in very brief, I tried to recap what was discussed in week 1, and I'll now quickly start with some questions that will help you solve the week 1 assignment. Okay, so please note, I am not going to tell you answers of week 1 assignment, but if you have any particular query to week 1 assignment, you can ask me. I'll be happy to help you, but I can directly cannot tell you the answers. Okay, so let's look at the first question. Obviously, you guys have to answer. Name 2 existing cell-based therapies that are currently being used in clinics. Anyone? So what do you understand by cell-based therapy? Let me give you some hints in order to understand this question. What do you mean by cell-based therapy? Cellular therapy, like anyone? Any idea? What do you mean by cell-based therapies? So you're trying to achieve a therapeutic result using cells. That is cell-based therapy. Means you're not using drugs, you're not using an individual protein, rather the cells are used as whole. Okay, a large number of cells are used to bring about a therapeutic effect in a particular patient, right? Now think which of the cell-based therapies you know are generally used in clinics and hospitals on a daily basis. Anyone? Okay, answer this. How many of you have given blood or donated blood? Or have you have heard of blood donation drives going on in your neighborhood or somewhere? Yes, no?
Yes, ma'am.
Yeah. So people donate blood, right? So what blood is composed of? I know most of you are from life science background, so it's an easy question. Even the non-life science people can tell us. What does blood have?
Cells.
WBC, RBC, and platelets.
Yes, yes. So blood contains cells. So can you say that blood transfusion is a cell-based therapy, right? No? Do you guys agree? It will be great if you guys interact. Otherwise, this session becomes very boring for me. Similarly, yeah. Similarly, people also give platelet transfusion. Have you guys heard platelet transfusion?
No, madam. I know the
No?
blood cells, different types of blood cells.
Yes. So when there is a like need of blood in surgeries, like people lose lot of blood, right, due to accident or something like that, so the volume of blood decreases. So in that case, people take blood from the blood bank of the same blood group and donate it. Similarly, in people who have a particular condition where the clotting doesn't stop, right, because of which continuous clotting doesn't happen, because of which continuously blood is losing from the body, in those cases people routinely give platelet transfusion. Platelets can trigger the clotting cascade and allow the formation of blood clots. Similarly, there is also bone marrow transplantation. Here, what happens? Transplantation. Here, what happens? So, it is not generally done to routinely, like to routine people. This particular treatment is for those who are undergoing or are going to undergo some chemotherapeutic treatment, like they have some cancer, right, and they are now going to undergo some chemotherapy or radiotherapy. So these kind of harsh chemicals that is injected in chemotherapy, it basically destroys everything in the body, right? And what people do before patient undergoes chemotherapy, doctor remove some volume of bone marrow from the patient and store it. Okay, so once the patient is done with the chemotherapy and needs fresh cells in order to revive its immune system, and it needs healthy blood cells, so in that case people then take out the patient's stored bone marrow cells and re-inject it into that patient, so that when the bone marrow is re-injected, it will like it is directly injected into the blood. The bone marrow cells, it can go and home at its current location where the bone marrow is supposed to grow. It will grow there, and the hematopoietic stem cells and then mesenchymal stem cells, these will start forming and help recovery of the patient. So that is called bone marrow transplantation. The cells are taken previously before the chemotherapy, and after the chemotherapy is done, doctors re-inject that bone marrow cells into that patient, so that the patient immune system and other blood cells and all, they all starts forming, new formation takes place, and the patient can recover better. So these are some examples of cell based therapies that are currently being used in clinics. Okay, so this is another question for you guys. Is my screen visible? The second question, are you guys able to see?
No, madam.
Okay, now
Yes,
is it now is it visible?
yes, it's yes.
Okay, so the question says that a group of doctors suggested to use beta islet cells from a cadaverous
No, ma'am.
pancreas. Yes,
Second
sorry,
question is
can
not visible.
hello?
Yes. No, it's changed.
Okay, so this is the second question. A group of doctors suggested to use beta islet cells from a cadaverous pancreas to address insufficient release of insulin in a diabetic patient. So, in which cell source category would you place these beta islet cells? Allogenic, autologous, syngenic, and all of the above. So, let me repeat the question. The question says that there's a diabetic patient, and the diabetic patient is in need of insulin. So the group of doctors suggested that you should take beta islet cells from a cadaverous pancreas. Cadaverous pancreas means from a recently dead body. Okay, from that pancreas you take some beta islet cells, and you use those beta islet cells to inject into the diabetic patient and basically address the insufficient presence of insulin. So the cells that are taken, this beta islet cells that is taken from the cadaverous pancreas, under which category of cell source would you place it? Will these cells be called allogenic, autologous, syngeneic, or all of the above?
Allogenic.
Allogenic.
Genic, right? So I think it's pretty obvious. But if since we are very large in number, still I would like you to understand why did you say it's allogenic for the sake of everyone's understanding.
Autologous is when the cells are derived from the own body. Allogenic when it is derived from same species but different body. And syngenic is when you derive it from someone's twin.
Yeah. So you're correct. So the answer is allogenic because the cells are derived from the same species but a different person, right? Now this is another question. Now, which of the following organs that is listed A, B, C, D is known as the body's most prolific organ? Skin, liver, bone marrow, or kidney? Anyone? Yes, someone has unmuted themselves. S-
Madam, I think liver. Skin. Madam, I think
Okay,
liver.
liver. One is saying liver, another is saying skin. Okay, now you guys can mute yourself. I request you guys to please mute yourself if you are not going to say something. Okay, if you are going to say something, then only you unmute yourself and speak. So we got two answers from the audience. There is kidney, sorry, hand, right, liver and skin. Someone is saying liver and skin. So actual answer is bone marrow. I'll tell you why. Bone marrow uh, resides the stem cells, mainly the hematopoietic stem cells and mesenchymal stem cells, and they are known to have very very high uh, division, like division rate. So within 12 hours it can regenerate around millions of cells. That is why bone marrow transplantation treatment is very popular. Even if you inject few 1 million or 2 million cells, within 2 3 days you will get 300 400 million cells. Okay, so that is why bone marrow is the most prolific organ in the body. After bone marrow comes I guess skin, right? Skin comes. So how the skin proliferates? Our topmost layer of the skin sheds itself every 28 days. So every 28 days we get a fresh coat of a barrier. Okay. Why this happens? Because the skin is composed of layers. Okay. The dermis layer and epidermis layer. So dermis layer is basically your fibroblast cells and the collagen and blood vessels are there. But the epidermis layer, which is the layer just above the dermis layer, it consists of different types of cells. So the bottommost layer of the epidermis consists of some stem cells. They routinely divide, okay, and differentiate and provide some cell population that migrate to top layers, and sequentially over a period of 28 days these cells come at the top of the skin barrier, all right, and when they come at the top they are dead, all right, and after 28 days these dead cells shed off. So skin is the third most prolific organ. And here, liver actually doesn't have a very good regeneration property. There is a treatment which is done where if a person is having liver failure and is need of liver transplant, so a relative with which the matching is done and most closest to it, they can give a half of its liver, and both the patient survive with half liver each. But the recovery time is very long. It can take like minimum of 1 year for entire liver to grow. And also there is one more issue. If you take out some healthy adult liver cells out from the body and try to grow it in lab, it is not possible. It will not grow well in vitro. So it's very difficult to work with liver cells. So people are trying to make pluripotent stem cells, and from that liver cells people are trying to make. Also, since human liver cells does not grow well in petri dishes or in cell culture, so they are using animal liver cells in order to do some experiments. Also, some cancer origin liver cells are also used. Then also kidney is not as prolific as the bone marrow and skin. So that is why the answer here in the four option is bone marrow. Okay. Now all is fine. You're trying to make tissue. I get it. But what is tissue, right? You have to first understand the biology of tissue in order to make a tissue. So that is what we are going to discuss in this particular question. So we have to fill in the blanks. Uh, Tissue is a collection of dash and dash that perform a given function. So you have to fill in the dash. So A says cells and extracellular matrix. B says cells and signaling molecules. C is signaling molecules, extracellular matrix. D is cells and biomaterials.
Let's see. Signaling molecules and extracellular matrix.
Okay. Anyone else would like to try?
Option A,
Okay.
cells and extracellular.
Okay. So we got two answers, I think. Anyone else would like to try? Okay. So the correct answer is cell and extracellular matrix. So a tissue, any tissue for that matter, like whether it's skin, kidney, liver, it is basically composed of two main things: the cell of that particular organ type, whether it's skin cells, liver cells, and cells need something solid onto which it can adhere and grow and spread. Okay, so that is the work of extracellular matrix, to provide support for the cell growth. Apart from that, ECM or solar matrix plays other functions as well. We will come to it later. But these two are the main component of a tissue. So that is why I told you the three triads of the tissue engineering, like what the three ingredients that you need to make a tissue is cell. Among the three ingredients, you need cells and ECM. We'll come to signaling molecules later. Okay, why signaling molecules are needed, from where they are coming from. But initially, from the birth, from when we are born, we are given with these two things: cells, right? And cells secrete ECM molecules, by the way, and create their own matrix, and in turn this particular ECM regulate cellular function. So it's a very dynamic environment that is there in a tissue, and that is why it's very complex for us to mimic that in vitro. Okay, so this is a very similar question that we saw earlier. Before the use of recombinant insulin started, diabetic patients injected porcine insulin into their bloodstream to normalize their glucose level. So the source of this insulin is allogenic, autologous, xenogenic, all of the above.
Xenogenic, ma'am.
Xenogenic. Yes.
Option C.
Yes. So you guys are correct. It doesn't need any more explanation. So yeah, we'll move on. Now, how do cells interact with the surrounding ECM components? Cells interact through receptors called integrins. There is a cell ECM interactive force that helps. Cells can interact through growth factors, or cells do not interact with ECM at all. What do you guys think? If anyone has read a bit of cell biology previously, maybe they can answer it.
A, ma'am.
Option
A.
A.
Okay. So that's great that few of you have been able to answer it. So those of you who don't know, for them, briefly I'll discuss what is why answer A is correct. So as I told you, cells, they secrete ECM molecules. Okay. And these ECM molecules are generally some proteins, but they are very large molecular weight, and because of which the diffusion capability of these protein molecules are very less. So some examples of ECM molecules are like collagen, then fibronectin, etc. Now, these particular ECM molecules, they have some particular sites present in them. So these are very large globulin kind of proteins. Okay. So they have sites for binding to growth factors. Then they have also some special sites that is called cell binding sites. Okay. And these cells, how will they like interact with these sites? Now, cell contains some receptors that are present on its surface. Now, these receptors are called integrins. These integrins are also themselves are proteins in nature. So, and proteins are made up of amino acids, and there's a particular sequence of amino acids that is unique, right? So these cell binding sites also have some unique set of amino acid present in them. Some example is like RGD, arginine. I think G stands for glutamic acid. uh, like just have a look of full form of RGD. So these are three amino acids which are generally found in the cell binding sites. And these RGD sites, they can be recognized by these integrin receptors. Okay. And these integrin receptors are themselves connected to cytoskeletal elements. Okay, so there are microtubules and all which are present inside the cells. So this cytoskeleton help in uh, maintaining some cellular morphology. They also play some important role in cell migration. Then you are able to understand these integrins are able to provide signals for morphology, for maintaining cellular morphology, as well as it provides signal for migration. So cells which are themselves are secreting ECM, this ECM is indirectly or directly, you can say, directly controlling cellular function like morphology, migration. Sometimes it also can help in cellular proliferation as well. If suppose these RGD sites are not there, cells will not adhere on our substrate. Okay, so and if you are trying to make the cells adhere on the substrate, through other receptors, then also it will not be possible because these integrins are connected to cytoskeleton. Okay, so any other receptors will not do the job. So in this way, cells and ECM, they interact through integrin, and this particular binding can indirectly affect many other processes, cellular processes as well as ECM related processes. Okay. Yes, this is the next question. So you guys have to answer which of the following questions are, which of the following options are true and which are false. Option A says the autologous chondrocyte transplantation causes donor side morbidity. Is it true or false?
True.
Sorry, I didn't get it. Can you repeat?
True.
It's true, ma'am.
True. Yes,
True.
it's true. So what happens in autologous chondrocyte transplantation? Anyone? Those who are saying that it's true?
In cartilage, we will be taking some cells, and again we will be replacing for it's majorly for the joints where we gonna replace. It will be from our own body cells in the sites of chondrocytes, and then we will culture it, and then we can uh, able to mimic the extracellular matrix. Basically, we can add into our own body system, ma'am.
Yes. So you are correct. Let me tell it again. So here what we are doing, we are taking some chondrocyte cells from the non-load bearing site. Okay. And we are taking it out so that we culture it to some number, like say 1 million cells. And then we take those cells, we can either grow the cells on some collagen matrix or directly inject the cells, like either way is possible, and we inject it where there is like more pain, maybe at the load bearing site. So so this procedure is like gives relief to the patient in the fact that it helps healing of the site where there is more pain, basically at the load bearing site. But in doing so, it also causes pain at the site where it was not meant to. Means we're artificially creating a wound that is called donor side morbidity. So this is called autologous chondrocyte transplantation. So now this is the second option we have. iPSCs cannot be used for transplantation. Do you guys think it's right or wrong? So what do you mean by iPSCs, first of all?
Induced pluripotent stem cells.
Yes. Now, do you guys think these cells can be used for transplantation?
They can be used, ma'am.
Yes, definitely it can be used. Rather, it's more easier to use iPSCs than using stem cells from another patient. Here you can take differentiated cells from the patient, and if you know how to uh, trigger the pathways that changes the programming of the cells to become stem cells, then you can easily use convert it into stem cells or induced pluripotent stem cells. And now these stem cells can again be signaled to differentiate along a pathway of your requirement, and it can be transplanted. Or you can directly transplant these stem cells and at the site. Now I have another question. Okay, which is better for transplantation purposes? To transplant differentiated cells or to use stem cells?
Stem cells.
Okay, why?
Stem cells can
can be used. They are not differentiated, so they don't have any specific function, so they can be used to make different types of cells.
So there is one disadvantage as well, right? Like since they are not programmed yet to differentiate along a specific lineage, if the in vivo environment is such that it gives some signals that is not of our requirement, that stem cells can like wander off into maybe becoming a cancerous cells. So again, my question still remains, which is better, uh, using stem cells or differentiated cells? So this is not correct actually, yes or no, like use only stem cells or use only differentiated cells. It is more on the application, for what application you are thinking. But I want to understand your point of view. If you are to ask to make an artificial tissue, which cell source would you opt for and why?
So it's better to use induced pluripotent stem cells or induced pluripotent stem cells or stem cells in vivo to generate the differentiated cells, and then we can transplant it to the individual. So maybe
So
like
you
that.
meant in vitro, not in vivo.
So to produce in vitro and
Yes, yes.
then become the differentiated cells, then we can be transplanted. So
Yes.
maybe like.
Yeah, I get you. So what she's trying to say is like you take the stem cells first, and since stem cells have a higher ability to division, like they can divide into a large number of cells, but the differentiated cells, they do not have that capacity of high cell division. So you take the stem cells, you divide it, you get the desired number of cells, and then you differentiate it into a specific lineage, and then you can transplant. So in that way, stem cells are more beneficial than the differentiated cells. But if you're not have the time to wait, right? So in case of autologous chondrocyte transplantation, we are actually transplanting differentiated cells. Chondrocytes, they are having a specific function. So we are directly taking the differentiated cells. We are not going to culture it for different passages and increase the cell number. We are just going to remove the ECM and get the individual cells out through trypsinization, all right? And we get a cell suspension, and when it's ready, we inject it. So in those cases, when you don't have the time or the resources to cause differentiation, you don't know the pathway through which differentiation is possible, then better to use differentiated cells because they're already specialized. You don't have to do anything to bring that stems bring the cells function, right? They're already functionalized. So depending on your application or what results you are looking for, you should accordingly choose the cell type. That is what the takeaway from this discussion is. Then the third option for this particular question is Integra is a commercially available FDA approved skin graft. Is it true or false?
True.
It's true. So Integra is actually a dermal substitute of skin. So I told you there are two layers of skin, epidermis and dermis. So the epidermis is the layer that contains the cells called keratinocytes, and dermis contains the cells fibroblast. So in burn patients where there is third degree burn, means the damage is up to dermis layer, all right? So your dermis layer is also compromised. So in those cases, integra like skin grafts can be used to replace the dermis layer first. And why replacing dermis layer is first important rather than go for the epidermis? Because dermis layer is the layer where all the important functions of the skin, whether it the blood circulation or the sensation of feeling, like you when you touch your skin you feel, right, something. So that nerves are present in the dermis layer. Then also the muscles are there, and like hair follicles are there, sweat glands are there. So the dermis is much more functionally important. Okay,
Hello,
so
madam.
we want, yes.
Madam, screen is not visible.
Is it not
Display is not
visible to?
visible. No,
Uh,
madam, your screen is not visible. The laptop
okay.
screen is not visible.
What about others? Can you guys see the screen?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Oh. So why don't you try to rejoin the meeting again? Because others are
Okay,
able
madam.
to see the screen. Okay.
Okay, madam.
Okay. Thank you. So as I was saying that dermis is like functionally important for us. So we have to first replace the dermis layer. We have to regenerate the dermis layer. And once that layer is healed, we can seed some keratinocytes onto a scaffold and then replace onto it. So similarly, there are other skin grafts available, like which are containing only epidermis specific cells, or which are acellular grafts are also there, which are just a polymer membrane, but they are biocompatible polymer membrane, so that gives a substrate only. Their cells are not there, they are just acting as a substrate onto which the host cells are allowed to migrate onto and grow. So those kind of skin grafts are also there. So anyone of you like aware of any other type of tissue engineered products that is available commercially, maybe looked up after the session, like after the water watching the lectures? Maybe you guys have looked up, or maybe remember from the discussion of the week one.
Infused bone graft.
Infuse. So what is
Yeah, I
that?
think
What is that? What is infused bone graft? I know Professor Vignesh discussed it, but let's recap. What was it? What was infused bone graft?
Hello, madam. Screen is not visible. Whether other candidates can able to see the screen?
Um, okay, so let
no, sir.
me reshare the screen. I think others are able to see the screen, I think. But let me
Now, madam,
reshare.
it's visible.
Now visible, ma'am.
Is it visible now?
Yes, madam.
Yes, madam.
So I was asking, since this problem is sorted, so I was asking, what do you mean by infused bone graft? Yeah, someone said that infused bone graft is an FDA approved tissue engineer product. So what is it? What are the ingredients in that particular product where it is used?
In dentistry.
Okay, okay.
Spinal fusion surgery.
Hmm. Okay. So the spinal fluid surgery is correct. But what is it? What are the things required to make it?
Bone morphogenetic protein, and the carrier matrix.
What is that matrix?
Carrier matrix.
What is the nature of that matrix?
Like it serves as a scaffold for bone formation. The bone morphogenetic protein is basically delivered in a collagen sponge matrix. So that's
Okay. Okay. So what is the biomaterial of that? What is the name of the material that is used as a scaffold? So these things you guys should start learning or picking up from the lectures. Tissue engineering is all about the types of materials that you can use, the types of cells that is available. So you should like start picking up those names, all right? It's important for you, so that later on it will be much easier for you to understand. if suppose you were to apply this particular subject elsewhere in your research, when you read research papers, you will be able to relate better. So infused bone graft, they were using a collagen based sponge, all right, which was like loaded with the recombinant bone morphogenetic protein. And when this particular spongy matrix with the signaling molecule BMP is used in surgery, the host cells, here the host cells are osteoblast, right? So these bone cells or osteoblast, they can migrate into the sponge like structure. And since BMP itself has the ability to uh, like cause bone formation, means they can signal the osteoblast cells to secrete collagen, then hydroxyapatite, and it can also allow the cells to trigger the cells to divide. So these processes like cell division, ECM secretion, right? So these processes starts, and that is like the process of healing, right? In short word, it is also called osseointegration. Maybe afterwards you guys can look up what is osseointegration. Osseointegration nothing is but new bone formation, all right? So when the bone cells come and integrate with the material that you have provided externally, that is called osseointegration, and due to which new bone formation happens, means more and more collagen and hydroxyapatite based ECM gets secreted, more bone cells start residing at that particular area. So that is how it is helping in bone regeneration. Here we are not trying to repair, rather we are trying to regenerate bone for a particular gap of like spinal cord. Okay. So that was one example of FDA approved commercial product, but there are many others. So why don't you guys, like after this session, I would request you to maybe Google it, all right? And like have a look what all products are there. Because in future, if you guys were to research, it is the first thing that you have to look after, like what is already there. So better to start now and increase your general knowledge in this particular area. Okay. So the next question is a match the following type of question. So read the question. I'll give you 2 minutes to read it, and then I'll ask what the answer you guys think. Okay.
A2B4.
A2B4. Okay. So let me mark it. A2B4. What about C and D? Also, whoever is answering, they have to justify as well later on. I'll ask why it is the correct answer they think.
D3, ma'am. D3.
D3. Okay. So that leaves C1. So let's go one by one. Okay, we have matched it. So first we have to understand why this is actually this is correct, by the way. This is the correct answer, but we have to understand why it is. So anyone, what is a natural polymer? What is a natural polymer? Anyone? What is the difference between natural and synthetic polymer?
Natural polymer means it is naturally available, like vegetables.
Means you're trying to say that it is available from a natural source, like
Yes,
animals,
ma'am.
plants, right?
Yes.
Whereas synthetic polymers, we make it in lab, right?
Yes.
So the chemistry of the synthetic polymers is much more defined, whereas the natural polymers, since we derived from the natural sources, we often see batch to batch variation. Although we know the chemistry of natural polymers, but the ratio of different groups, the polymer weight and all, those are not very much controllable. Whereas in synthetic polymers, we can control which atoms to add, how many how many like bonds should be there. Like it's very, very controlled and fine tuned. The molecular weight is very much controlled by us. So that is the difference between natural and synthetic polymers. So under natural polymers, here is the correct answer is chitosan. So anyone know from where chitosan is derived? Since it is a natural polymer, it must be derived from a natural source. But from where it is derived? Anyone knows?
Insect shell, no?
Yes, can you repeat?
Shells of the insect. I'm not sure.
Insects. You said insects. Actually, it's not insects, it's actually the Cretaceous fishes like the prawns and the crabs. So their shells, the outer shell, they have a very hard outer shell that is called exoskeleton. Very hard exoskeleton is there. So that is made up of a protein called chitin. So when this particular protein is processed, acid based processing is done, then we get a polymer that is called chitosan. Okay, so it is not directly derived, but we have to process chitin in order to get chitosan. Okay, and also chitosan is very, very widely used. It has excellent antimicrobial properties, very good mechanical strength. So but it may have some limitations. I have not personally worked with chitosan, but I know people who worked with it. But since this is a non-veg product, so if you are making any like I know someone in our lab who are making chitosan based food coatings, right? So in order to increase the shelf life of products. So since this is a non-veg based product, so you may have to declare that it is non-veg. So yeah. So, but apart from that, for tissue engineering application, chitosan is very widely used. Then next is hydroxyapatite. So hydroxyapatite is nothing but a compound made up of calcium and phosphate. All right. So it is inorganic and it is mixed with the organic phase that is collagen. And these two are the main components of bone ECM. That and because of chitosan, bone is very stiff in nature. All right. Then I'll come to PEG later. First, cobalt chromium alloy. So, as the name suggests, it is a metal alloy. All right. And metals are generally used as for orthopedic as well as dental applications. Why? As I told you, bones are having very high Young's modulus. In order to match that particular strength, people are using metal based biomaterials like this cobalt chromium alloy. All right. So, in order to make screws, plates, staples, these kind of surgical things that goes directly on that is used to fix broken bones. So, any other example of alloys similar to cobalt chromium alloys that you have read previously or recall from week one lectures? Anyone?
Nitol.
Hmm? Can you repeat?
Nitina.
Yes, that's correct. What it is made up of? It's a commercial name, by the way.
Nickel and titanium.
Yes, that's correct. Any other example? So stainless steel can also be used for orthopedic implants. Then titanium based alloys, but with other metals like titanium, magnesium, vanadium. So these kind of alloys are also there. Also, please note the stainless steels that we use in our homes to make utensils like through like the plates and the bottles, that grade of titanium, that grade of stainless steel is not used. Okay, that is 304 grade, I think. And for orthopedic
306L.
huh?
306L, madam.
I think 304. I think so, 304. Okay,
Okay,
and but for orthopedic applications we use 316. That is also low carbon L. Okay, so there are different grades of stainless steel that is permissible for using orthopedic in that can be used for biomedical applications like for orthopedic implants. Okay, then I left out one option, that is C, polyethylene glycol. So it is a synthetic polymer, all right, and it's a bio-inert polymer. First, any one of you, let me know what do you understand by the term bio-inert.
Material which could not promote bone ingrowth.
Not bone growth, means cells cannot interact with that particular material. Okay, so if you have this particular material inside your body, the immune cells and all, they will not be able to recognize it. Okay, that is called bioinert, or a biological response won't be visible to this particular material. I'll discuss briefly why it is bioinert in nature, this particular material, why it is bioinert in nature. So as I told you, cells, in order to interact with the substrate, what they need some sites, cell binding sites, right? That is, for example, RGD sites, because it is the integrin receptors present on the cell surface which can recognize these particular sites. So first thing is, this particular polymer is not going to have that cell binding sites, which is why cells cannot recognize it. Also, many synthetic polymers which are used for tissue engineering application onto which cells are grown, they they can have various degree of wettability properties. So what I just said, I said wettability properties. Means the surface of the polymer can either be hydrophobic or hydrophilic. Hydrophilic. Hydrophilic means water loving, hydrophobic means it repels water. So this PEG or polyethylene glycol, they are hydrophilic in nature. They strongly bind water molecules to itself. Whereas hydrophobic polymers are also there, like PCl, polycaprolactone. So they are having very high contact angle value. Okay, so they are hydrophobic in nature. So what happens is, when you're trying to select a biomaterial for growing cells, we try to maintain a balance between hydrophilicity and hydrophobicity. We do not want the material to be very hydrophilic in nature or very hydrophobic in nature, because either scenario, in either scenario, cells will not be able to interact. So in whereas in case of polyethylene glycol, it is highly hydrophilic. Okay, very hydrophilic in nature. Because of this high hydrophilic nature, the cells are not able to interact with this polymer, and that is why it is bioinert. That is why it is called bioinert polymer. Okay, did you get why PEG is bioinert? Because it is highly hydrophilic. It is water loving. It has no cell binding sites present. Okay. It also cannot allow any adhesion, like through hydrophobic forces. It cannot even allow adhesion of some proteins onto its surface. So if suppose some proteins get adhered through hydrophobic or van der Waals forces, then also some cell protein interaction can take place on the biomaterial surface. But since it is highly hydrophilic in nature, it does not permit protein adhesion as well. That is why it is bioinert. Okay, any doubt in this particular matter? Yes, Janki, you have a query? Anyone has any query? So is this point clear? Why PEG is a bioinert polymer? If it is not clear, feel free to ask. I won't mind to repeating it again. But these small things you should know, because tissue engineering is a very complex subject. You guys should know bit about biomaterials, bit about cell biology, then bit about signaling as well. So like a combination of various disciplines is there. So little bit knowledge, general knowledge is required for understanding the subject better. Any doubt in understanding, or if there is anyone who didn't understand why PEG or polyethylene glycol is a bioinert polymer? Is it clear? Maybe a thumbs
Understood.
up would work. Understood. Okay. I got one response. Great. So I'll assume that rest of you have also assumed. Okay. So this is another question I have. Cell signaling using growth factors, it can be categorized under. So you have to select multiple options. It can be of endocrine nature, paracrine nature, autocrine, or all of the above. Anyone? There can be more than one correct answer here. So let me repeat the question again. You can achieve cell signaling using growth factors, and what are the, so how would you categorize the cell signaling using growth factor? Whether it will fall under endocrine signaling, paracrine signaling, autocrine signaling, or all of the above. Again, those who have studied cell biology or cell signaling previously as a course, maybe in your bachelor's, you may be able to answer it.
Option B and C.
Option B and C. Okay. So you're correct. It is B and C. So those of you who couldn't understand the what is endocrine, what is paracrine, what is autocrine, let me briefly explain. So endocrine signaling, it happens for hormones. So it's a signaling, um, like hormones, they are secreted by endocrine organs like pancreas or adrenal glands. So these hormones, they are secreted directly into blood vessels. So the source of hormone and the target, they are very far apart. And the hormones, they travel using the blood to reach the particular um, site, target site. So why it is possible? Because these particular hormone molecules, they have a longer half-life. They are stable for a longer period of time. Okay, that is why endocrine signaling is possible. Whereas growth factors by nature, they are very small protein molecules with a very short half-life. So if we were to think that suppose they were to transmit like transported from one location to another through blood vessels, it will not be able to reach a very far target organ, which is why signaling through growth factors only happens in a paracrine fashion or an autocrine fashion. So here paracrine signaling, what happens? There is a signaling cell, okay, and there is a target cell. Now the distance between the signaling cell and target cell is very less, all right? Maybe neighborhood cells it can be, okay? So this signaling cell releases some growth factor molecule, and these, since these are small molecules, they can diffuse, all right? Diffusion can take place, and diffusion is concentration dependent. As you know, concentration is higher near the signaling cell, and as the target cell's location is going further and further, less molecules will reach. So depending on how many growth factor molecules were able to bind to the surface receptors of the target cell, you will get different intensity of response in that particular target cell, right? So that is called paracrine signaling. Whereas autocrine signaling can also happen. The signaling cell can itself signal itself. So these growth factor molecules which are released with signaling cell, they can bind to the receptors that is present here, okay, on the surface of the signaling cell. And this is very like general that happens. So in cases of tissue formation, like morphogenesis, this happens. Then when there's signal for cell division and there's a need for a very large number of cells to start the cell division cascade altogether, then paracrine, autocrine signaling both can happen altogether. So these are the two pathways that the growth factor molecules uses in order to do cell signaling. Okay, endocrine signaling cannot take place. Now, this is another question. Which of the following strategies are useful to hide foreign organs, sorry, foreign cells from body's immune system? Select all that apply. So, you know, foreign cells, they can cause immune rejection, right? So, what strategies can you use to prevent immune rejection or immune response to foreign cells? There are options like use your basic knowledge or general knowledge that you have gained from week 1 and try to answer it. We can encapsulate the foreign cells inside a bioinert material. We can stop the expression of IHC or immune histocompatibility complex on the surface of cells using genetic engineering. We can directly inject the cells at the target size, or all of the above. Anyone? Which of the following
Maybe
strategy? Yes, yes.
option A, ma'am. Encapsulate foreign cells inside a biomaterial, bioinert material.
Okay. So anyone else would like to try? One person is saying that option A is correct. Anyone else? Do you guys agree with her? So we are trying to basically make sure that the foreign cells may be allogenic or xenogenic origin. Those cells, they survive inside our body. Okay, in order for it to survive, we have to protect it from our body's immune system. So how would you protect those foreign cells so that it can do its function for which they are delivered, okay, and without getting killed by the immune cells? So one person is saying that we can encapsulate it inside a bioinert material. Any other opinions on this? So what about option B? Does it looks like it can be a useful strategy to do? Or what about option C? What would happen if you directly inject the cells, the target site? It will anyway reach, right?
But there's a chance of immune rejection in the option C.
Okay, that is why you're not saying that option C is correct. Okay, agreed. What about option B then?
I don't think so, ma'am. I'm not. I don't know,
Okay,
ma'am, about that.
okay, no problem. Anyone else? Yes, you have raised your hand. You can uh like unmute yourself. Rijuana P. Go ahead. You have raised your hand. If you have something to say, you can unmute and respond. Rijwana Praveen, yes, you can unmute yourself and respond. You have raised your hand. Apparently, he forgot that he has raised his hand. Anyone else would like to try? Devika has answered what she thinks is correct. What about others? It's a very general question, right? You are going to artificially make a tissue. So the cell source may not be directly from the patient every time. It may be from a different source. So in that case, how would you protect the cells such a way that when you deliver those cells, they are protected and is able to do the function for which you have transplanted those cells? How would you make sure? I think up to whatever we have discussed, I think for most of you, option A seems correct, right? Like Devika said. If you guys don't interact, I won't be able to understand how much you have understood or which which point should I discuss, right? So it's essential for you to interact, ask questions.
B can also be correct. Since if we suppress the immunohistochemistry of a cell, it will not be recognized by our immune system.
Yes. So I think you're correct. So B can be also a useful strategy to do. Okay, but if you directly inject this as a target site, maybe for a few hours the cells can do its job, but it won't be for a very long period of time because nearby immune cells will be triggered and they will be immediately targeted for killing. So the IHCs or the immunohistocompatibility complexes, they are present on the cell surface, and that is what the immune cells uses to recognize whether the cells are its own origin or from somewhere else. Okay, so if we like somehow stop the expression, right, of these IHC complexes altogether, so these cells now become they do not have a identity for the immune cells to recognize. So these are also called universal donor cells. People are also working in the universal donor cells, although it is not yet commercially available. But these universal donor cells do give us some hope that we can still inject cells from different sources if we know how to make these universal donor cells without worrying about immune rejection, but we still don't know on doing it what more problems we are going to create. So that is why this approach, option B, is not very common yet, but approach A is widely used. We like use islet cells, right? Beta islet cells. You must have read that in patients whose beta islet cells are compromised, and in order to like reduce frequent intake of insulin, why don't we inject the beta islet cells? So for doing it, we can encapsulate into a biomaterial like PEG is a bionut biomaterial. So we can encapsulate the beta islet cells taken from a healthy patient and encapsulate it and then transplant it, so that for a sustainably long period of time, these cells can secrete insulin. Now, the encapsulation should be such that there is a continuous flow of oxygen and nutrients from the host body to the foreign cells. But it should not allow the entry of immune immune cells. So we have to encapsulate in that way. So there should be some degree of porosity. We can play with the porosity of our polymer. That porosity should be small enough for the diffusion of oxygen and other nutrient molecules like glucose and all, but it should not be too big that it can allow the entry of antibodies or antigen presenting cells. So in this way we can like optimize with the properties of our biomaterial in order to encapsulate foreign cells and successfully deliver it inside our body for a period of time that it can do some therapeutic benefit to the patient. Okay. Now, these are some examples. These are 4 examples of tissue engineered products or biomedical products that are available in the market. Now, I want you guys to answer in which circumstances these products will be useful for a patient. Like if you were a biomedical engineer and someone asks you like like I have a product in which patient to use. So case A, case B. So you have to answer the disease or the condition that the patient have for which this particular product is useful. Okay. So first is extracorporeal liver devices. So these are some liver devices. So if you are not familiar with, let me briefly explain first. So these are artificial liver, you can say, okay, where the liver cells or the hepatocytes, they are cultured on a biocompatible biomaterial, like a porous biomaterial through which nutrients and oxygens can diffuse, but immune cells cannot get in. And as you know, liver has many secretory properties as well, right? So the liver cells can secrete all the important useful products, and those products can be diffused out from that device. So it's a artificial device. It is designed um, to function for a short period of time. It is not a permanent solution. So in which patients would you think these extracorporeal liver devices would be useful?
Ma'am, acute liver
Liver cell.
failure.
Yes, yes. So it
Hepatitis.
will be, yes, it will be useful for those patients whose liver has stopped working, and maybe they are waiting for a new liver, means maybe they are waiting to get a new liver from a donor. So for the period of time they are waiting, the doctor can recommend that you use this extracorporeal liver device in order to sustain for a period of time that you have, right? Then artificial skin graft, we already discussed one example of it. So in which circumstances like an artificial skin graft be useful?
Skin damage.
Very, very vague term, skin damage. What kind of skin damage? So I have a cut in my arm, say, so a graft will be useful.
third-degree burns.
Yeah, for burns, right? Acid burns, fire burns, right? Also, also it is useful in those patients who are having chronic wounds. So are you guys aware of wound category called chronic wounds, where the wound does not heal on its own? Okay, so it's very common in patients with diabetes. Their like wound healing capacity reduces because of very high glucose level in their body. So they frequently get chronic wounds and places, and that requires interventions like continuous medication. And if you don't protect that wound site well, it will get infected. So in those cases, in order to, so since skin is a barrier, right, of our body, and that protects our internal soft organs. So if that barrier is compromised, then it is very easy for the foreign infectious agents to get inside and like kill us. So that is why in those cases as well, for chronic wounds as well as for burn victims, artificial skin grafts are useful. You can also use it for aesthetic purposes, like for plastic surgeries and all, where people themselves want to change some aspect of their particular body, and like they want something replaced. So for that purposes as well, skin grafts can be useful. So let me write burn wounds or chronic wounds. Then intraocular lenses. So these lenses are not contact lenses. Let me remind. So these are the lenses which helps us to see, but these are artificial. So in which cases these intraocular lenses will be useful? Or for which kind of patients these lenses are designed?
Those who are suffering with cataract.
Yes, you're correct. Cataract. Okay, so in old age, when people get cataract, means their natural lens, they get some very foggy, they are not able to see. The transparency is reduced in those lenses. So the doctors replace those natural lenses with intraocular lenses surgically. Okay, and that helps the person to see. So these lenses are very biocompatible and very widely used. Okay, then total hip joint replacement when it is useful.
Damage of joint.
Damage of joint,
Joint, ma'am. Hip joint.
hip joint.
Yes.
Did you say hip joint? So what
Yes,
kind of
ma'am.
damage are we talking about?
Means ball and socket breakdown.
Yes, yes, the ball and socket joint that is connecting your pelvis and your femur. If that joint gets broken, suppose, so in it is very common in old people who have a very low bone density. They have very less strength in their bones. So accidentally, if suppose they fell and break their hip joint, so nothing can be done. Like in those cases, the patient cannot does not even have very high regenerative capability because the patient has become very old. So in those cases, they replace the entire joint, natural joint, with the artificial hip joint. Okay. So these are, so this question helps you to understand what all products are there available in the market that are designed to make the quality of life better for us. Okay. So tissue engineering is a very, very powerful field, and almost every other institute in our country has few researchers who are working on tissue engineering or some aspect of tissue engineering. And like as and it doesn't matter if they have biological background or not, the material scientists are working, mechanical engineering people are working, because it's such an important field that has the potential, to alleviate the pain from so many patients all around the world. It's very nice that you guys have registered and for this course, and I want you guys to like learn as much as possible through this course. Okay, if you have any particular interest in knowing more about the research, I request you guys to go through literatures, research papers that is available, right, along with the course content. You will understand better. So if you are looking for any PhD positions later on in this field, it will definitely help you to realize which area of research in tissue engineering would you like to pursue. So this is another question that I have. Select the biomaterial from this list. Very easy. So there is penicillin, orthoscrew, metformin, thermometer, which is a biomaterial here. Anyone? Okay, just quickly, what do you understand by the term biomaterial? Biomaterial, right? So what do you understand by that term? Sir, like in the lecture videos, sir has very briefly discussed this term biomaterial. Later on, he will get into more depth, but like from that understanding only, I want you guys, like I want to know how much have you understood the term biomaterial? Anyone?
Metamorphism.
I'm not asking the answer right now. I'm asking what do you mean by biomaterial? So for tissue engineering, or one of the ingredient to make an
The
artificial
material
tissue
which
is
is
biomaterial.
composed of cells.
Yes, can you repeat?
The material which is composed of cells.
Okay.
Biomaterial is a natural or synthetic material that are placed into our body which perform the damaged tissue organ.
Okay, fine. Very simple definition. By the way, biomaterial does not have to have cells in it. Okay. The other answer that someone gave, I think you have looked up in the Google. If
No, madam.
no, okay, fine. Anyone else would like to try? What do you understand by the term biomaterial?
Substances which can interact with
Materials
our
are implanted into human body.
Okay. And once, like someone else was also speaking, I think he was interrupted. You can speak now.
substances which can interact with our biological system.
Okay. So you guys are like together. If I say together, the answer is correct. Individually, the answer is not complete. So biomaterial, the purpose of a biomaterial is to artificially mimic the ECM, the extracellular matrix of our tissue. Agreed? Because it is not possible for us to get ECM directly from our body, right? That is why we are trying to replace that artificially. That is where the biomaterial comes. So if I were to define what is biomaterial, it's a material which is like implanted into our body and shows biological response, biologically favorable response. So there's a term called biocompatible. Okay. So people generally say biomaterial should be biocompatible. Means if you are using a material, say PEG, polyethylene glycol, for suppose drug delivery, so it should deliver drug. It should not cause a side effect of say um, bacterial infection. If it is doing that, if it is or because of that you are seeing bacterial infection happening at the site of delivered drug, then it is not biocompatible. So a material, if you're implanting inside the body, it should be biocompatible as well. Now, please note, drugs are not biomaterials. Okay, drugs are not biomaterials. Again, I'm repeating. So, given the understanding so far, what you guys have told and what I have told, now you answer. Which of the following in this list is a biomaterial?
Option B.
Option B. Yes. So someone was saying that metformin is a biomaterial. Now is it clear why metformin is not a biomaterial? It is actually a drug. Metformin is a drug for diabetic patients. Penicillin is also a drug. And thermometer, it does not invasively gets inside our body. It's a device, right? Used to measure the body temperature. So it is not a biomaterial. So ortho screw, which is usually made up of the metals that we talked about, like titanium, stainless steel. So these kind of alloys or metals that are used to make these ortho screws, these are biomaterials. So this question is very easy. If you understand what is a biomaterial, then you will be able to answer this kind of conceptual questions. Okay. Any doubt, anyone? Okay, I'll move on. Okay, that was the last question for the today's class. If you have any particular doubt regarding the lecture content or the assignment, I'll be happy to take it up. Or if you have any query related to questions that I've discussed in 1.5 hours, you can also ask me. The floor is open for your questions. Yes.
Actually, I have a doubt, ma'am.
Yes, go ahead.
Ma'am, yes, ma'am. You in lecture you have given 2 weeks. The assignment should be submitted on which day, ma'am? For week 1 and week 2.
So I think the last dates are mentioned. So week 1 assignment has to be submitted before 7th of August, week 1 assignment. Okay. And
Yes, ma'am.
for week 2 assignment as well, you have to submit by 7th of August. So what they generally do in NPTEL, they give you more time to do assignment in the previous weeks, like week 1 and week 2, because many people start watching the lectures delay maybe, or join the course late. That is why they have kept very uh, big deadline, means they're giving you more time to solve the assignments. But I'll discuss week 2 content in the next week, because I think 7th August, no, so you guys must be completed with your week 2 assignments as well. So in the oh, third August is our next session. Sorry. So in 3rd August also you will have enough time to go through week 1 content and week 2 content. So deadline for both week 1 and week 2 assignment is the same. That is 7th August. And you guys have time now to go through the content.
Okay, ma'am. Ma'am, and I also have other doubt. Like I have registered the course and done my payment. So I need to do any allotment for seat or anything
No,
for
no.
credits?
So you are going to give exam, that's why you have paid, right? So they will
Yes,
inform
ma'am.
you what you have to do. So towards the end of the course they will give you a hall ticket, admit card, something like that, for you to go to a center where you have to give exam. And once you like get above 50%, I think, and as well as in assignments also, you have to cross 60 to 70%. I'm not very clear about the minimum marks that you have get, please check it, but you have to cross that threshold marks in order to get the certificate. So study well for this course in order to get very good marks. Okay, they
Yes,
will
ma'am.
inform you,
Ma'am,
NPTEL people will inform you. Yeah.
Okay. And another one last doubt, ma'am. For each lecture, for example, one, I have a questions over there, and I'm submitting the answers, and it's showing again the same questions, like a new one. Should I need to answer, or how will I get to know whether I have answered those questions, whether the people have taken it or not? I'm not getting that, ma'am.
So you have posted that question where?
So down, I used to do in the laptop. Usually, I
Okay.
used to see the videos, and down of the session, you will be having some 5 to 6 questions. And I'm giving the submit button. For next time, I open the same video. It's showing the same answers, questions. Like I have not answered it. How is it, ma'am?
So it is just a self-assessment type of a
Okay.
question. It is there. They are that is not marked, by the way. That is like to help you revise the content. So and like if you watch this video even after 2 weeks, you'll be able to have this question. So don't
Exactly.
worry about it. Okay. If you have
Yeah.
answered it, you can check the answer, right? It will give you what is the
Yes. Yes,
correct
ma'am.
answer. Yes. So it's just for self-assessment. It is not going to be graded. Don't worry.
Okay, ma'am. These
Hello,
were the
madam.
queries. Thank you, ma'am.
Yes.
Madam, whether I can download the lectures,
Whether you can download the lectures. So
which is given uh, portal, in the portal
mhm.
for tissue engineering, in the course, content, weekly content, the lectures are given. Whether I can download those lectures,
So I
then I
am
need not
not sure.
study putting
Yes.
internet connection in my
Uh,
laptop.
s-
Then I need not
yes.
study putting internet connection in my laptop. In offline also, I can access the content.
Yes, yes, I get it why you want to download it. I actually don't know how to do it. Okay. You can ask this directly to NPTEL. You can drop a mail to NPTEL and they will help you and they'll let you know whether it is downloadable or not. And since these videos are available on YouTube, YouTube doesn't consume a lot amount of data, by the way. Like I would
Okay, madam.
ask you to download the videos in YouTube app rather. So you can go to YouTube, you can download the entire playlist there in your mobile or in a laptop at one go. You don't have to rely on the NPTEL website. You know, right? It is available on YouTube as well, the playlist, the video playlist. So you can download all the videos in the YouTube app.
Okay, madam. Madam,
Yes.
actually, there is not much written content in the lectures. Which slides have shown are in the in the lectures. So, if I get the lectures of those videos, then it is easy. Otherwise, actually, I have a habit to read the contents. Actually, here in this course, the written content is not providing.
Yes, actually it is provided. So let me show you my screen. So if you go here, the course outline, you can see there is a option called download videos and there's a download now thing. And in
Oh,
order
okay,
to,
madam.
okay, and in order to get written content, there is a thing called books. If you go to English book and it will click here and you will be able to see the entire lecture, entire course basically. So here it is a transcript. The written content is the same as what the professor is saying in the lectures. So they have compiled it into a book manner so that you can read through it and revise the lectures by reading. So both things are available. Okay, so
Madam,
this
book
book,
is whether book is available in the NPTEL.
yes, yes, you can click here in the
I can
option here.
access the book, madam.
Yes, yes, you can download it. Yes.
Okay, madam. Actually, I thought I have to buy a book. I have to
No,
pay
no, no.
few
You
thousand
don't you
rupees.
don't need to uh, like do any expenses for this course. But additionally, if you want to learn apart from this course, I can recommend you additional reference books. But for this course and for the exam, whatever content that is there in this particular portal, like the videos, the transcripts and the assignments, that is enough for you to clear the exams. And as well as
Trans
I'm also here to help. So yeah.
Okay, madam. Madam, transcript is available in the portal. So I can written transcript. I can download the written transcript.
Yes, yes, yes. You can download it. It is also
Yeah,
downloadable.
okay, madam. Okay, madam.
Okay.
Okay, madam.
Yeah. So both things are sorted. Okay. Any other queries?
Ma'am, can you mention that reference books, ma'am, which you told now?
Okay. So I have studied tissue engineering from a book. So let me show you my screen. I'll Google it for you guys and give me a second. So let me share my screen. So there are multiple books available. So you guys can get this tissue engineering book by Palson and Sangeeta and Bhatia. This is a very nice book, very simple language that they have written. Other than that, there is a book by Robert Langer, Joseph Vacanti as well. So multiple editions are available of this book. And the Sangeeta and Palson, this particular book is not available in internet. Means its PDF version is not available in is internet, but you can get this book by Robert Langer and Joseph Vacanti easily in PDF form. It is available. All right, so you guys can download it. This book is bit advanced. Let me like say it's this Principles of Tissue Engineering by Langer and Vacanti. It is slightly advanced. So if it is not required, then don't get into it. But if you feel that you want to learn more, then learning is like never stops. You can read more and more. All right. So these two books I would recommend apart from the course content that is there.
Thank you, ma'am.
Okay. Any other queries? So all of you have seen week 1 content before coming to today's session, or you are going to see later now? So a small feedback maybe. Did you guys understood what I have discussed today? Would you like me to improve on something, or would you have like wanted more question types to be discussed, like any feedback from your side?
Ma'am, we understood the class really well.
Okay, that's great. Thank you. So I'll try to put more questions. All right. And any theoretical part that we didn't understand, let me know, like maybe in this week or next week. And don't worry that if that topic is not discussed in that week, you may not be able to ask. ऐसा कुछ नहीं है. You can ask me in week 3, week 1 ka content, no problem. Just study in your own pace. All right. And whatever doubts you have, feel free to ask. If you don't ask, the people who are there to support you, help you, won't be able to help you. All right. So you have to ask questions, then only you will learn better. All right. Okay. If there is nothing more to be discussed, then I'll close off today's meeting. If you have any more things, let me know. All right then. This recording will be made available to you soon. All right, and we will meet next week. That is on 3rd of August, where we'll discuss week 2 content. All right. Thank you all for joining.
Thank you, ma'am.
Yeah.
Thank you, madam.
Thank you, ma'am.
Yeah. Thank you.
Thank you, ma'am.
Thank you, madam.